What is meiosis? Meiosis is a type of cell division in sexually reproducing organs that results in 4 cells with half the number of chromosomes. It has 2 cell divisions, meiosis I and meiosis II.
Meiosis is the separation of homologous chromosomes. The first stage of meiosis I is prophase I. Here, Simran is showing the nuclear envelope of the cell breaking, and then Becca and Nithya are the centrosomes and their arms are the microtubules. Also, the chromosomes start to condense. Each chromosome pair, who are represented by Prag and Sim's arms, cross over, which is where the genes are swapped between the non-sister chromatids. As a result of crossing over, X-shaped regions form called the chiasmata. Later, in prophase I, microtubules, Becca and Nithya, join to the kinetochores of the sister chromatids. The second stage of meiosis I is metaphase. In metaphase I, pairs of homologous chromosomes, represented by Prag and Sim's arms, line up at the metaphase plate with one chromosome of each pair facing each pole. The microtubules of the mitotic spindle continue to attach to the kinetochores. The third phase of meiosis I is anaphase I. In anaphase I, the homologous chromosomes separate and move toward opposite poles. This happens because the ends of the kinetochore microtubules, represented by Becca and Nithya, begin to break down and pull the homologs apart. However, sister chromatids stay and move together as a unit. It's just the homologs that split up. The fourth phase of meiosis I is telophase I. In telophase I, each half of the cell has a complete haploid set of duplicated chromosomes, and cytokinesis occurs simultaneously. Cytokinesis is the division of the cytoplasm and will form a cleavage for own animal cells while forming a cell plate in plant cells. The nuclear envelope starts to form again and chromosomes decondense. Meiosis II is the separation of sister chromatids. The first phase of meiosis II is prophase II. First, the nuclear envelope breaks down. In prophase II, no duplication of chromosomes occur, but a spindle apparatus, represented by Becca and Nithya, forms again. Microtubules attach to the kinetochores. The second stage of meiosis II is metaphase II. In metaphase II, the chromosomes are all placed at the metaphase plate as a result of the microtubules pushing them. The third stage of meiosis II is anaphase II. In anaphase II, the sister chromatids, Simran and Prag, separate and are pulled to opposite ends of the poles as individual chromosomes. The fourth stage of meiosis II is telophase. In telophase II, the nuclear membrane reforms, which is represented by Sim and Prag's envelopes, and the chromosomes start to decondense, and the 2 cells start to split into 4 cells due to cytokinesis. The 4 cells are now haploid cells. Becca, Nithya, Prag, and Sim represent these haploid cells, which contain half the set of chromosomes.
Who's that chromosome I see over there? That's me and my sister chromatid. What's that spindle thing reaching my kinetochore? They're from centrioles. There's 2. I think meiosis every day, that I woke up as a diploid, and I can't help splitting again, and I need that cleavage to occur, If I was you, I'd love meiosis too. I'd love meiosis too. I'd love meiosis too. If I was you, I'd love meiosis too. I'd love meiosis too. I'd love meiosis too. I walk in as a diploid, I go straight to prophase. I always have to cross over my homologs behind me. Metaphases, microtubes, homologs attach opposite poles, woo! And even proteins try to separate homologous pairs. I take meiosis every day, that I split during telophase, and I can't help splitting again, and I can't wait to be haploid. If I was you, I'd love meiosis too. I'd love meiosis too. I'd love meiosis too. If I was you, I'd love meiosis too. I'd love meiosis too. I'd love meiosis too.